بسم الله الرحمن الرحيم. السلام عليكم ورحمة الله وبركاته. اتكلمنا المحاضرتين اللي فاتوا عن General Structure of the Gastrointestinal Tract، وعرفنا إن Gastrointestinal Tract is formed of esophagus, stomach, small intestine and large intestine. واتكلمنا بالتفصيل عن histological structure of the esophagus and histological structure of the stomach. إن شاء الله النهاردة هنبدأ كلامنا عن small intestine. The small intestine is divided into 3 segments. The first segment after the stomach is duodenum, then jejunum, then ileum. They have many common characteristics. What is the function of the small intestine? First, completing digestion, then selective absorption of useful substances. It also secretes some enzymes and hormones. What is the structure of the small intestine? The wall of small intestine consists of the usual 4 main layers, which are mucosa, submucosa, musculosa or muscularis externa, and serosa. The mucosa, the first layer from the side of the lumen, is composed of epithelium, lamina propria, and muscularis mucosa, as usual. But the mucosa of the intestine have certain structural characteristics to increase the surface area for absorption to about 200 square meters. What are these structural characteristics? The first one is Plicae circularis, villi, crypts, and microvilli. These structural characteristics help to increase the surface area for absorption. The first one, Plicae circularis. What is the Plicae circularis? There are mucosal folds with submucosal core. The mucosa form folds. These folds take a core from the submucosa. These Plicae circularis are numerous in the upper part of the small intestine, especially in the region of the jejunum. They increase the surface area for absorption by 3 folds. Notice here, the mucosa as a whole form folds and take core from the submucosa. Intestinal villi. What is the intestinal villi? They are mucosal finger-like projection. Their length about 0.5 to 1.5 millimeter long. Their side, they cover the mucosal surface in a large number, giving the small intestine a velvety appearance. What is the structure of the villi? Epithelium of the villi. The villus, as we said, it is a finger-like projection from the mucosa. So the epithelium of the mucosa project into the lumen of the intestine and take a core from the lamina propria and also some smooth muscle fiber of the muscularis mucosa. What is the type of epithelium present at the or line the villus? The epithelium is simple columnar absorptive epithelium, represent 90%. It has brush border, numerous microvilli. The second cell present in the lining of the villus is the goblet cell, which secretes mucus. The third cell is the enteroendocrine cell, which secretes intestinal hormone as secretin. After the epithelium, there is a core of connective tissue formed by the lamina propria. In all layers of or in all segments of the gastrointestinal tract, the mucosa is formed of epithelium and underlying it lamina propria, then muscularis mucosa. There are the three layers of the mucosa. When the epithelium form a projection or finger-like projection into the lumen of the small intestine, this finger-like projection is called villus. This villus is covered by epithelium. The type of cell present in this epithelium is simple columnar absorptive cell plus goblet cell plus enteroendocrine cell. Under the epithelium, as usual, there is a layer of loose connective tissue formed by the lamina propria of the mucosa. In this loose connective tissue, there are central lymphatic capillary and fenestrated blood capillary. The central lymphatic capillary, called central lacteal, which have an important role in the absorption of fat, and a rich network of fenestrated blood capillary that involve in the absorption of nutrients. After that, the smooth muscle cell extend into the villi from the muscularis mucosa. Contraction of these muscle cells increase the efficiency of absorptive process and help the central lacteal to empty their fat content into the larger lymphatic in the lamina propria. So, the function of the villi increase the surface area for absorption by about 10-fold. The third structural specification which is present in the intestinal mucosa is the intestinal crypt or the intestinal gland or the glands of Leiberkhun. What is the intestinal crypt? They are invagination of the epithelium into the lamina propria, forming simple tubular glands or crypt, extending almost to muscularis mucosa. What is the structure of the crypt? The epithelium of the villi is continuous with that of the crypt, and we add two cells. We said previously that the epithelium of the villi is formed of simple columnar absorptive epithelium plus goblet cell plus enteroendocrine cell. Now in the crypt there are the three cells plus Bennett's cell and undifferentiated stem cell. Now what is the function of the crypt? The presence of crypt adds to the increase in the mucosal surface area done by the villi. Now, when the epithelium of the intestinal mucosa projects into the lumen in the form of finger-like process, it is called villus. When this epithelium is invaginated into the lamina propria to the level of the muscularis mucosa, it is called intestinal crypt, or intestinal gland, or gland of Leiberkhun. So, the epithelium lining crypts is formed of 5 types of cell. Simple columnar cells with brush border, abundant microvilli, goblet cells, enteroendocrine cells, Bannis cell present in the basis of the gland, undifferentiated stem cell. The fourth specification, which is present in the intestinal mucosa, is the microvilli. Microvilli are present in the apical surface of the simple columnar absorptive cell. They are large in number. So the apical border is called striated or brush border. The function of these microvilli is to increase the mucosal surface and facilitate the absorption. We all know that the mucosa is formed of 3 layers: epithelium, lamina propria, and muscularis mucosa. And we said that the epithelium may project into the lumen and take a core of lamina propria and a part of muscularis mucosa forming a villus, or may invaginate into the lamina propria forming an intestinal gland or intestinal crypt or crypt of Leiberkhun. Now the second layer of the mucosa, which is the lamina propria. It is a layer of loose connective tissue containing intestinal glands. This layer is rich in lymphocyte, plasma cell, macrophage, eosinophil, and small lymphatic nodules. The small lymphatic nodule lie in the lamina propria. But if this lymphatic nodule become larger, it may extend to the submucosa. What is the lymphatic nodule? It is aggregation of blood lymphocytes. For example, in the ileum region of the small intestine, the lamina propria contain Byers patch. Byers patches are large lymphoid follicles. They may bulge through muscularis mucosa into the submucosa, opposite the mesenteric attachment. These Byers patches have immunological function. The third layer of mucosa, muscularis mucosa, as any part of the gastrointestinal tract, it is formed of 2 layers of small muscle cells, the inner one circularly arranged and the outer one longitudinally arranged. Submucosa. Submucosa, it is formed of loose connective tissue. In the region of the duodenum, it contains mucus-secreting glands called Brunner's gland. Musculosa, as any part of the gastrointestinal tract, it is formed of 2 layers of smooth muscle cells, inner circularly arranged and outer longitudinally arranged. The serosa, the fourth layer, the outer layer, it is formed of loose connective tissue covered by peritoneum. It covers jejunum, ileum, and most of the duodenum. At the beginning of the lecture, we said that the small intestine is formed of three segments: duodenum, jejunum, ileum. And we said that all the segment of the intestine is covered by villi. But the shape of the villas is differ from region to another. For example, in the duodenum, the villus is broad leaf-like, while in the jejunum, the villus is tongue-shaped and short. In the region of the ileum, the villus is finger-like and is absent over the region of the Peyer's patch. The second difference: in the region of the duodenum, the submucosa contains Brunner's gland, mucus secreting gland, while in the jejunum, the submucosa contains no Brunner's gland. In the ileum, the lamina propria contains Peyer's patch, which may extend to submucosa. The amount of goblet cell is less in the duodenum, more in the jejunum, and more numerous in the region of the ileum. Here there is the difference: the presence of Peyer's patch in the region of the ileum, lamina propria, and the presence of the Brunner gland in the submucosa of the duodenum. The jejunum contains no Brunner's gland in the submucosa, nor Peyer's patch in its lamina propria. Now we will discuss the histological structure of the epithelium of the villi. We said that the epithelium of the villus is formed of three types of cell: columnar absorptive cell, goblet cell, and enteroendocrine cell. The first type of cell, columnar absorptive cell. When we see the columnar absorptive cell by light microscope, they are tall columnar cell. They have oval basal nuclei. They have acidophilic cytoplasm, and they have apical striated or brush border due to the presence of abundant microvilli. By electron microscope, the apical surface of each cell has abundant long microvilli, about 3,000, covered by cell membrane, and has a core of actin microfilament. So, this microvilli is covered by the cell membrane of the columnar cell and has a core of actin microfilament. The digestive enzymes are located in the cell membrane of the microvilli. As any cell membrane, the cell membrane of the microvilli is covered by a coat of glycoprotein, which is called glycocalyx. This glycocalyx or the glycoprotein coat has a protective function and also provide binding site for a specific substance to be absorbed. The cytoplasm of these cells contains pre-ribosome, rough endoplasmic reticulum, smooth endoplasmic reticulum, well-developed supranuclear Golgi, and many mitochondria. There are two types of junctional complex present between epithelial cells. The first type is tight junction, and the second is adherence junction. The function of tight junction is to seal off the intercellular space. What is the meaning of seal off? Close firmly the intercellular space. And the adherence junction keeps the cell from pulling apart under tension, Pulling apart means separation. What is the function of the columnar absorptive epithelium? First, digestion. Proteins are broken by peptidase into amino acid and carbohydrates broken by disaccharidase into monosaccharide. Then absorption of digested protein and carbohydrate into the blood vessel and the fat into smooth endoplasmic reticulum and then to the lymphatic vessels. The second type of cell, goblet cell. They are unicellular mucus secreting gland, each cell considered as a gland. They are scattered between columnar absorptive cell, and as we said, small amount in the duodenum, more amount in the jejunum. It is most numerous in the region of the ileum. The expanded apical part is full of mucinogen secretory granules. The narrow basal part contains nucleus and cytoplasmic organelle. They produce acid glycoproteins that protect and lubricate the lining of the intestine. The third type of the cell, enteroendocrine cell. They are present in the epithelium of the intestinal villi and also in the basal region of the intestinal gland. These cells have the characteristic of diffuse neuroendocrine system. They need a special stain as silver to be seen. We see an example in the stomach region. There is enteroendocrine cell which is stained by silver, and we said that, or we call it as argentaphene cell. These cells are polypeptide secreting cells and containing basal secretory granules. There are many types of these cells which produce different peptide hormone. Some cells secreting secretin, other secreting cholecystokinin, other secreting pancreozymin, and other secreting endorphins. M cell or microfold cells. This type of cell particularly found overlying Peyer's patch, where there is no villi. What are the characters of M cell or microfold cell? They are broad cells with dome-shaped upper surface. These cells have no brush border. There are tight junctions present between the M cell and the intestinal absorptive cell at their free surfaces. The basal surface has a concavity between it and the basal lamina. This concavity accommodates lymphocyte and macrophage from the underlying lymphatic tissue. The basal lamina under the M cell is discontinuous, which facilitates transition between them and the lymphatic nodule in the lamina propria. These cells have a role in the immunological reaction of the intestinal mucosa. It acts as antigen-presenting cell, as they endocytose antigen and transport them to the underlying lymphocyte and macrophage, initiating immune response. This is the shape of M cell. Broad-shaped cell have dome-shaped and underlying concavity. We notice that the basal lamina underneath the M cell is discontinuous. The M cell acts as antigen-presenting cell. The epithelium of the crypt. We said that the epithelium covering the villi continue down to line the crypt along their upper parts. The bottom of the crypt contains two additional cell types. Beside the columnar absorptive epithelium, goblet cell, enteroendocrine cell, the base of the crypt contains two additional cell types, which are called undifferentiated stem cell and Bannis cell. Undifferentiated stem cell or crypt-based columnar cell. They are also columnar cell, but they have no brush border. They have no apical microvilli. These cells undergo mitosis to replace the cell of the villi and the crypts. There is continuous renewal of the intestinal epithelium of the villi every 3 to 6 days. What is the cell which is responsible for its replacement is the undifferentiated stem cell. Bannis cell. Bannis cells are present in groups at the base of the crypts. They are large cells containing abundant large acidophilic apical secretory granule and basal basophilia represented by abundant rough endoplasmic reticulum. So these cells have the characteristic of protein synthesizing cell. Their nuclei are rounded and basal. The function of these cells, they produce an antibacterial enzyme called lysozyme that controls the bacterial flora.